we could not find any mechanistic literature evidence, in PubMed for the estrogen receptor transcriptional regulation of STAR-D10. But what is known from the literature, however, is that pancreatic insulin content is actually regulated by the estrogen receptor. So if you take all of this information in total, and if you visualize STAR-D10, the estrogen receptor, and insulin as a three corners of a triangle, now the literature makes some connections, but SPP forms the third side of that triangle and allows the users to hypothesize a possible mechanistic basis for the ER insulin connection. And that hypothesis may that estrogen receptor is regulating insulin status in part due to its transcriptomic regulation of STAR-D10. All right, so sometimes these hypotheses can be difficult to generate if you only have a specific gene in mind. And so for this reason, we allow users to back their queries out, sort of zoom out, and encompass a broader range of cellular functions using Go terms. An example of hypothesis generation, again, of relevance to NIDDK, I'm going to pose here as an investigator seeking novel mechanistic evidence underlying the known link between EGF and brown adipose tissue formation. All right, so I'm going to go back to our live demonstration here, reset our filter, and we're going to enter in a Go term search, and here we're going to begin entering a couple of keywords to bring up brown fat cell differentiation. We'll select that. Now this serves as a proxy for a number of genes involved in brown cell fat cell differentiation. But know that on a run of a Go search, you need to specify the node class. And here we're going to select receptors. And this is to prevent potentially very large queries being submitted that might impact server performance. And so I've selected receptors and catalytic receptors. I'm going to click submit. And the default data display is the target display, meaning that these are all of the gene targets that were in the Go ontology term for brown adipose tissue formation that are also have evidence in SPP. Now, in order to narrow these down, what I can first do is I can select the category view. And what this does is now it breaks down those same data points into receptor families that may be involved. And so since I'm specifically interested in EGF receptors, I'm going to go back to our form, and I'm going to narrow down my search just to the EGF receptor family here and click submit. And so what this does is now I'm only looking at those brown fat cell data point genes, related genes that have something to do with EGF receptor, either down or upregulated in blue and red. And in the regulation report, I can see evidence for EGF receptor family regulation of PPAR gamma. Now, this is actually consistent with what is known in the literature, that PPAR gamma is regulated by EGF receptor family members. And so now this is actually kind of a positive control for my search, which gives me more confidence that the results I'm getting back from SPP are real, and that I can believe the other evidence that I see. So really what we really want to know is what are the novel EGF receptor family targets. And so what I want you to notice is that two different EGF receptor family ligands, TGF alpha and NRG1, both of these repress laminin 4 expression. Now, this is interesting in the context of evidence from the literature suggesting that laminin-4 is a negative regulator of brown fat tissue, of fat browning rather in mice. And so again, we have this complementary relationship between the literature and SPP, whereby the literature provides the outlines of the hypothesis and SPP provides evidence for the mechanistic basis of that hypothesis.